All right. Good afternoon, everyone.
Good afternoon, sir.
Good afternoon, sir.
I hope you're all very well.
Yes.
And the grace of God, we are fine.
We are fine.
All right.
And you?
All right. Nice to meet you. All right. So let's get on with serious business and see what we can do. I think last time we were able to meet and we had a very lengthy discussion. Actually, it was an introductory meeting. So I introduced you to the course, shared a few things with you, and then we started with our first topic. I attempted to define biochemistry to all of you. And I explained to the effect that biochemistry is defined as one, an aspect of biomedical science that explores the nature and biochemical composition. The nature and biochemical composition of living organisms. And number two, the interaction that occurs between these components. The interaction that occurs between these
Hello.
Yes. And then thirdly, hi, the influence of these interactions on the structure, physiological function, and general well-being of living organisms. So in essence, this is what we did. And I hope you all remember this definition. So I said that usually, as an outline, we'll begin with the biochemical composition. The biochemical composition is what we term as biomolecules. And then the interaction that occurs between them is what we call metabolism. And then the influence on the structure, the physiological function, and the general well-being. This is where we talk about clinical microbiology or medical microbiology, clinical biochem, clinical biochemistry, or medical biochemistry. Are you okay? So in essence, this is what we did. So we started off with biomolecules. So we said biomolecules. It consists of what? All the biochemical components. All the biochemical compounds that occur within our cells or body. The emphasis is on what? All the biochemical compounds. So there are so many. They are diverse and ubiquitous. So what it means that they are of different forms. And then they are also widely distributed through our body. That's why we say they are diverse and they are also what? Ubiquitous. And because they are diverse, it will be very difficult to take them one by one. So for ease of our discussion, we attempted to group them into two. So we said that there are two groups.
Hello, sir.
Hi.
Sir,
Please, I'm
please,
listening.
I'm unable to see the handwriting.
So
I'm unable to see the handwriting, please.
that's why I'm talking. Are you okay? So you can hear me and make meaning. So
Okay.
we said that they can be
Yes,
grouped into
please.
inorganic biomolecules. And then we have what? Organic biomolecules. There are three types of the inorganic biomolecule. We have water, we have the gases, primarily carbon dioxide and oxygen. And then we have the minerals. These are you okay?
Yes, please.
Yes, sir.
Yes, sir.
All right, so this is what we did last week. I'm sure you remember. So we looked at the water and we spoke at length about what it is, why it's important in humans, and then where water can be found in our body. There are three main locations in our body where water can be found. Number one, they can be found in what? Inside cells. In fact, the largest proportion of the water in our body are within cells. So about 70 to 80% of the water in our body are within cells. Then you have some also within fluids. Water serves as the basic component of fluids. So here I'm talking about the extracellular fluid and the intracellular fluid. Okay. The intracellular fluid is what makes up about 70 to 80%. The extracellular fluid consists of what? Interstitial fluid and intravascular fluid. And then water also occurs inside various tissues. Are you okay? So even in the skeleton, where we expect the bone cells to be very calcified and relatively dry, it still contains some reasonable amount of water. Okay. And how do we acquire water? Physiologically, there are three main ways by which our body acquires water. Number one, through rehydration. So when you take in water or any drink, are you okay? That water, even though part comes out as urine, part is also retained within our body, all things being equal. Number two, we acquire water through the food we eat. So when you are taking the palm nut soup, the porridge, the beverage, they all contain water. And through
Water.
metabolic
Yeah, water.
so through metabolic processes, some metabolic reactions generate water at the end of the day, like oxidative phosphorylation. During respiration, this generates a lot of water. Are you okay? And these water molecules, yes, part may be lost through breath, but some may also be kept in the body. Because the body temperature is not too hot. Is I okay?
Sir, please, can you take it again?
Take what?
Yes, sir. The last statement.
I said that water can be acquired within our body by three main ways. Number one, through rehydration. So when you are taking water or anything that contains water, so the drinks, the fruit juices, and then the water itself. Then the food we eat, the porridge, the soups, and etc. And then through metabolic reactions. Example is the oxidative phosphorylation, during which water is formed. Is that okay?
Yes, sir.
Yes, sir.
Yes, sir.
All right.
Yes,
Any
sir.
question? Any question?
Sir,
Hi, dear.
please, is the respiration the same as the oxidative phosphorylation?
Oxidative phosphorylation is an aspect of respiration. Respiration involves two main reactions: oxidative phosphorylation and electron transport chain, ETC. Okay. Usually during electron transport system, water is produced as a final product.
Okay, so thank you.
You're welcome. Okay. So today we are starting off with what? Organic biomolecules. Are you okay? I also recall that we spoke about the gases, carbon dioxide and oxygen. And I told you that while oxygen is very important for energy generation, are you okay? Carbon dioxide is also very important for maintaining the pH of body fluids. And this is no joke. If the pH of your blood changes by just one factor, the whole blood may cease to even flow well. And there would be dire clinical outcomes. So carbon dioxide comes in very handy. It's going to dissolve in the water in blood and form what you call carbonic acid. So it helps to buffer the blood fairly constant through changing scenes of time during the day and all that. So this is what happens. On the minerals, there are two. We have the metals and the non-metals. Examples of the metals include what? The calcium, if you remember. So the minerals, we have the metals and the non-metals. The metals are calcium, selenium, zinc, potassium, sodium, the non-metals are chlorine, iodine, and etc. So, and there may be phosphorus. Then you have fluorine. These are all common examples of non-metals which occur within our cells.
Sir.
And they have hello.
Hello, sir. Please.
Hi.
Are we taking the organic or inorganic?
The
Because
inorganic. The
okay,
inorganic.
so we are done with the organic because I'm confused.
No, I haven't started yet.
Okay.
We did this
Thank you.
the first time we met, and I felt that some of you may not have joined. That's why I'm just recapping. So if it's a bother to you, I can end it here.
No, sir.
No,
I
sir.
don't understand where
No.
we were going.
No, sir.
Because
Sir, no.
it's organic.
So can I continue?
Yes,
Yes,
please.
sir.
Yes, sir.
All right. So we spoke about the minerals and we said that there are two kinds, metals and what? Non-metals. The metals are largely calcium, selenium, zinc, potassium, sodium, etc. Yeah, I think magnesium as well, and others. There are many of them. Even copper. Yes. And then the non-metals consisted of chlorine, phosphorus, iodine, fluoride or fluorine, and then Yeah, I think these are the major ones. There are others, but these are very major. So they make up the non-metallic minerals. And these are very vital to health, general. And so I would want to leave this to you to go read about it. Find out the physiological role of these minerals within the body. Sodium and chlorine are very important, and potassium are very important in nerve impulse transmission. Nerve impulse transmission and muscle coordination.
How do you go here? So show you
All right.
like this.
Okay. Please, can we start off with the organic biomolecules?
Sir,
Yes,
please.
sir.
The
Yes,
nerve
sir.
impulse. Sir, please. The nerve impulse transmission and the muscle coordination. Some benefits of the metals and non-metals.
No, I was saying that the sodium, the potassium, and chloride ions are very important in nerve impulse transmission and muscle coordination.
Thank you.
You're welcome. We know what impulse transmission is and why it's very important. Without it, you lose your sense of awareness. Your whole body will be numb. You can't even see anything. You can't even blink your eye. So it's very important. Then selenium and zinc are very important in developing the immune system. So they boost the immune system and make it robust. That's why we give zinc tablets to people who have infection. It doesn't kill the pathogen. It's not an antibiotic. But when you take zinc tablets, they strengthen the immune system so that the immune system will be able to fight the infection. That's why they administer zinc, not particularly for any infection, but for almost all infections, because it's not specific. Its target is not the pathogen, but rather what? The immune system. So it strengthens the immune cells, whether it's T lymphocytes or whatever. Now the next one is going to be what? Okay. So I'm going to show you a short video on carbohydrates, and then after that, we'll look at the organic biomolecules, and then we'll continue. Are you ready?
Yes, sir.
Yes, sir.
Yes, sir.
Have I showed you any video before?
No, sir.
No, sir. No, sir.
Oh,
Okay.
okay. All right, so let's watch.
Series as a part of McMaster's Demystifying Medicine. I'm Matt.
And I'm Susie.
And today we'll be focusing on carbon dioxide and the chemical role it plays in our bodies to keep us alive. Have you ever wondered why a person would suffocate if he or she is deprived of oxygen and exposed to a large amount of carbon dioxide? Well, you will by the end of this video.
So for today's agenda, we will be talking about the influence of carbon dioxide, the clinical disorders of acid-base homeostasis, and a recap of the presented information.
Did you know that a person suffering from diabetes has acidic urine and a higher breathing rate than a normal person? Why is that? Not surprisingly, you're not the first person to wonder. Since the latter half of the 19th century, many researchers have tried to find the reason behind this phenomenon. One of these researchers was Friedrich Walter, who gave dilute hydrochloric acid to a non-human mammal and observed that his breathing rate increased.
The search for the answer didn't end here. With progression in the field of chemistry came new knowledge. In 1907, researcher Lawrence Henderson became the first person to place the role of carbon dioxide on a theoretical basis, thereby modifying the law of mass action, as described in the previous video. He described the role of carbon dioxide as shown below. Carbon dioxide in water makes carbonic acid, which dissociates into protons and bicarbonate. From this discovery, Henderson was instrumental in establishing the Henderson's equation, which helped to measure the acidity of a solution as shown below. The concentration of protons in a solution is equal to K, a unitless constant multiplied by the ratio of weak acids to its conjugate.
Henderson later modified his equation to this, as he knew that the concentration of carbonic acid depends on the pressure of carbon dioxide. This equation is not only important mathematically, but also conceptually, as it relates the acidity of the solution to the level of carbon dioxide. In this equation, the concentration of protons is equal to K, again, another unitless value, multiplied by the ratio of the pressure of carbon dioxide to the concentration of bicarbonate. Thus, if pressurized carbon dioxide is halved, the acidity of the solution will increase unless the concentration of bicarbonate is lowered. It must be remembered that this relationship is an equilibrium, so the reaction can go in either direction. Both the arteries and veins help maintain this equilibrium by carrying and removing oxygen and carbon dioxide respectively. This helps maintain homeostasis.
Ever wonder how homeostasis is examined in our body? It's actually quite simple. In any routine clinical practice, there are three components in our blood plasma that are used to measure our body's homeostasis. 1. The total concentration of ions from strong acids like sodium and chloride, which are the two most important ions in our blood plasma. 2. The total concentration of weak acids, which, as mentioned in our previous episode of How the Body Works, are the amino acids, protein molecules of the hemoglobin, and phosphates. And lastly, 3. Carbon dioxide, which is often calculated using the concentration of bicarbonate. When all three criteria are met, homeostasis is achieved and all is good to go. However, in the unfortunate case when homeostasis is not achieved, clinical disorders may result. These disorders are commonly known as the following: respiratory acidosis and alkalosis, named after the decrease and increase of the physiological pH of 7.4 respectively due to the imbalance in the concentration of bicarbonate, and metabolic acidosis and alkalosis, known for the decrease and increase of pH respectively due to the imbalance in the concentration of strong ions and weak acids.
However, things are not as simple as defining disorders by the irregularities in pH in the body. As you might have already imagined, the body likes consistency, and has many ways to adjust for the imbalance of pH. This is referred to as adaptive responses. Let's consider hyperventilation, for example. When a person hyperventilates, there is an increase in the partial pressure of carbon dioxide in the body, which results in respiratory acidosis. To compensate, that individual would excrete chloride in one's urine to cause an increase in strong ion difference. This change in ion concentrations causes an increase in bicarbonate in the bloodstream, which can be defined as metabolic alkalosis. As you can see, while acidosis and alkalosis can be defined by a change in pH, there are, in fact, many other physiological and metabolic changes taking place in the body that characterize this change. Also, there are modifications that subsequently occur to lessen the severity of the imbalance. Many major illnesses in the body result in these changes in acid-base homeostasis, and each variable has to be carefully treated so the body can return to normal.
To conclude, there are three mechanisms to control blood pH: the carbon dioxide pressure system, balance of strong ions, and plasma proteins. It is safe to say that the carbon dioxide pressure system is remarkable for its ability to change rapidly with breathing. Hence, it's a rapid contributor to the control of homeostasis in our blood. But hold on, didn't we mention something regarding strong ions and essential plasma proteins in our previous video? Where did they go? Fortunately, the kidneys also play a vital role in homeostasis. However, in comparison with the carbon dioxide system, the rate of control for the concentration of strong ions and plasma proteins depend on the excretory function of the kidneys, which could take several hours. Therefore, keeping a constant pressure of carbon dioxide in our blood is highly crucial for our survival. But at the same time, we must appreciate the beauty of carbon dioxide and how it makes our sodas taste so good.
Well, that's all we have for today, folks. Thank you guys for listening, and we hope you enjoyed today's episode of How the Body Works. Feel free to click, follow, and subscribe. Check us out on Facebook. or visit our website at www.demystifyingmedicine.ca. See ya!
Bye, guys!
Welcome to yet another exciting episode of How the Body Works, an educational series as a part of McMaster's Demystifying Medicine. I'm Matt.
And I'm Susie.
And today we'll be focusing on carbon dioxide and the chemical role it plays in our bodies to keep us alive. Have you ever wondered why a person would suffocate if he or she is deprived of oxygen and exposed to a large amount of carbon dioxide? Well, you will by the end of this video.
So for today's agenda, we will be talking about the influence of carbon dioxide, the clinical disorders of acid-base homeostasis, and a recap of the present.
All right, so having gone through it, shall we all discuss it shortly? What did you pick up?
Hello, sir.
Hi there.
Yeah, please, at the beginning of the video, I think it was talking about how humans are able to use oxygen and then bring out carbon dioxide for the environment to also use, and how important oxygen is to human beings.
Was it how important oxygen or carbon dioxide?
Carbon dioxide.
Okay. So what did he mention as possible application of carbon dioxide to the benefit of humans? I think somebody raised their hand, so let me talk to her. Grace. Hello, Grace. Hello. Yeah, Elizabeth.
Sir, please, blood
Can we rewatch the video again?
blood homeostasis.
All right. Just watch this shortly. For those of you who may want to watch it, please listen. Now note that if I show you videos, it's not just for
Did you know
entertainment.
that a person suffering from diabetes has acidic urine and a higher bleeding rate than a normal person? Why is that? Not surprisingly, you're not the first person to wonder. Since the latter half of the 19th century, many researchers have tried to find the reason behind this phenomenon. One of these researchers was Friedrich Walter, who gave dilute hydrochloric acid to a non-human mammal and observed that its breathing rate increased.
The search for the answer didn't end here. With progression in the field of chemistry came new knowledge. In 1907, researcher Lawrence Henderson became the first person to place the role of carbon dioxide on a theoretical basis, thereby modifying the law of mass action, as described in the previous video. He described the role of carbon dioxide as shown below. Carbon dioxide in water makes carbonic acid, which dissociates into protons and bicarbonate. From this discovery, Henderson was instrumental in establishing the Henderson's equation, which helped measure the acidity of a solution as shown below. The concentration of protons in a solution is equal to K, a unitless constant multiplied by the ratio of weak acids to its conjugate.
Henderson later modified his equation to this, as he knew that the concentration of carbonic acid depends on the pressure of carbon dioxide. This equation is not only important mathematically, but also conceptually, as it relates the acidity of the solution to the level of carbon dioxide. In this equation, the concentration of protons is equal to K, again, another unitless value, multiplied by the ratio of the pressure of carbon dioxide to the concentration of bicarbonate. Thus, if pressurized carbon dioxide is halved, the acidity of the solution will increase unless the concentration of bicarbonate is lowered. It must be remembered that this relationship is in equilibrium, so the reaction can go in either direction. Both the arteries and veins help maintain this equilibrium by carrying and removing oxygen and carbon dioxide respectively. This helps maintain homeostasis.
Ever wonder how homeostasis is examined in our body? It's actually quite simple. In any routine clinical practice, there are three components in our blood plasma that are used to measure our body's homeostasis. 1. The total concentration of ions from strong acids like sodium and chloride, which are the two most important ions in our blood plasma. 2. The total concentration of weak acids, which, as mentioned in our previous episode of How the Body Works, are the amino acids, protein molecules of the hemoglobin, and phosphates. And lastly, 3. Carbon dioxide, which is often calculated using the concentration of bicarbonate. When all three criteria are met, homeostasis is achieved and all is good to go. However, in the unfortunate case when homeostasis is not achieved, clinical disorders may result. These disorders are commonly known as the following: respiratory acidosis and alkalosis, named after the decrease and increase of the physiological pH of 7.4 respectively due to the imbalance in the concentration of bicarbonate, and metabolic acidosis and alkalosis, known for the decrease and increase of pH respectively due to the imbalance in the concentration of strong ions and weak acids.
However, things are not as simple as defining disorders by the irregularities in pH in the body. As you might have already imagined, the body likes consistency and has many ways to adjust for the imbalance of pH. This is referred to as adaptive responses. Let's consider hyperventilation, for example. When a person hyperventilates, there is an increase in the partial pressure of carbon dioxide in the body, which results in respiratory acidosis. To compensate, that individual would excrete chloride in one's urine to cause an increase in strong ion difference. This change in ion concentrations causes an increase in bicarbonate in the bloodstream, which can be defined as metabolic alkalosis. As you can see, while acidosis and alkalosis can be defined by a change in pH, there are, in fact, many other physiological and metabolic changes taking place in the body that characterize this change. Also, there are modifications that subsequently occur to lessen the severity of the imbalance. Many major illnesses in the body result in these changes in acid-base homeostasis, and each variable has to be carefully treated so the body can return to normal.
To conclude, there are three mechanisms to control blood pH: the carbon dioxide pressure system, balance of strong ions, and plasma proteins. It is safe to say that the carbon dioxide pressure system is remarkable for its ability to change rapidly with breathing. Hence, it's a rapid contributor to the control of homeostasis in our blood. But hold on, didn't we mention something regarding strong ions and essential plasma proteins in our previous video? Where did they go? Fortunately, the kidneys also play a vital role in homeostasis. However, in comparison with the carbon dioxide system, the rate of control for the concentration of strong ions and plasma proteins depend on the excretory function of the kidneys, which could take several hours. Therefore, keeping a constant pressure of carbon dioxide in our blood is highly crucial for our survival. But at the same time, we must appreciate the beauty of carbon dioxide and how it makes our sodas taste so good.
Well, that's all we have for today, folks. Thank you guys for listening, and we hope you enjoyed today's episode of How the Body Works. Feel free to click, follow, and subscribe. Check us out on Facebook, or visit our website at www.demystifyingmedicine.ca. See ya!
Bye, guys!
Welcome to yet another exciting episode of How the Body Works, an educational series as a part of McMaster's Demystifying Medicine. I'm Matt.
And I'm Susie.
And today we'll be focusing on carbon dioxide and the chemical role it plays in our bodies to keep us alive. Have you ever wondered why a person would suffocate if he or she is deprived of oxygen and exposed to a large amount of carbon dioxide? Well, you will by the end of this video.
So for today's agenda, we will be talking about the influence of carbon dioxide, the clinical disorders of acid-base homeostasis, and a recap of the presented information.
Did you know that a person suffering from diabetes has acidic urine and a higher breathing rate than a normal person? Why is that? Not surprisingly, you are not the first person to wonder. Since the latter half of the 19th century, many researchers have tried to find the reason behind this phenomenon. One of these researchers was Friedrich Walter, who gave dilute hydrochloric acid to a non-human mammal and observed that his breathing rate increased.
The search for the answer didn't end here. With progression in the field of chemistry came new knowledge. In 1907, researcher Lawrence Henderson became the first person to place the role of carbon dioxide on a theoretical basis, thereby modifying the law of mass action, as described in the previous video. He described the role of carbon dioxide as shown below. Carbon dioxide in water makes carbonic acid, which dissociates into protons and bicarbonate. From this discovery, Henderson was instrumental in establishing the Henderson's equation, which helped measure the acidity of a solution as shown below. The concentration of protons in a solution is equal to K, a unitless constant multiplied by the ratio of weak acids to its conjugate.
Henderson later modified his equation to this, as he knew that the concentration of carbonic acid depends on the pressure of carbon dioxide. This equation is not only important mathematically, but also conceptually, as it relates the acidity of the solution to the level of carbon dioxide. In this equation, the concentration of protons is equal to K, again, another unitless value, multiplied by the ratio of the pressure of carbon dioxide to the concentration of bicarbonate. Thus, if pressurized carbon dioxide is halved, the acidity of the solution will increase unless the concentration of bicarbonate is lowered. It must be remembered that this relationship is an equilibrium, so the reaction can go in either direction. Both the arteries and veins help maintain this equilibrium by carrying and removing oxygen and carbon dioxide respectively. This helps maintain homeostasis.
At Ever wonder how homeostasis is examined in our body? It's actually quite simple. In any routine clinical practice, there are three components in our blood plasma that are used to measure our body's homeostasis. 1. The total concentration of ions from strong acids like sodium and chloride, which are the two most important ions in our blood plasma. 2. The total concentration of weak acids, which, as mentioned in our previous episode of How the Body Works, are the amino acids, protein molecules of the hemoglobin, and phosphates. And lastly, 3. Carbon dioxide, which is often calculated using the concentration of bicarbonate. When all three criteria are met, homeostasis is achieved and all is good to go. However, in the unfortunate case when homeostasis is not achieved, clinical disorders may result. These disorders are commonly known as the following: respiratory acidosis and alkalosis, named after the decrease and increase of the physiological pH of 7.4 respectively due to the imbalance in the concentration of bicarbonate, and metabolic acidosis and alkalosis, known for the decrease and increase of pH respectively due to the imbalance in the concentration of strong ions and weak acids.
However, things are not as simple as defining disorders by the irregularities in pH in the body. As you might have already imagined, the body likes consistency, and has many ways to adjust for the imbalance of pH. This is referred to as adaptive responses. Let's consider hyperventilation, for example. When a person hyperventilates, there is an increase in the partial pressure of carbon dioxide in the body, which results in respiratory acidosis. To compensate, that individual would excrete chloride in one's urine to cause an increase in strong ion difference. This change in ion concentrations causes an increase in bicarbonate in the bloodstream, which can be defined as metabolic alkalosis. As you can see, while acidosis and alkalosis can be defined by a change in pH, there are, in fact, many other physiological and metabolic changes taking place in the body that characterize this change. Also, there are modifications that subsequently occur to lessen the severity of the imbalance. Many major illnesses in the body result in these changes in acid-base homeostasis, and each variable has to be carefully treated so the body can return to normal.
To conclude, there are three mechanisms to control blood pH: the carbon dioxide pressure system, balance of strong ions, and plasma proteins. It is safe to say that the carbon dioxide pressure system is remarkable for its ability to change rapidly with breathing. Hence, it's a rapid contributor to the control of homeostasis in our blood. But hold on, didn't we mention something regarding strong ions and essential plasma proteins in our previous video? Where did they go? Fortunately, the kidneys also play a vital role in homeostasis. However, in comparison with the carbon dioxide system, the rate of control for the concentration of strong ions and plasma proteins depend on the excretory function of the kidneys, which could take several hours. Therefore, keeping a constant pressure of carbon dioxide in our blood is highly crucial for our survival. But at the same time, we must appreciate the beauty of carbon dioxide and how it makes our sodas taste so good.
Well, that's all we have for today, folks. Thank you guys for listening, and we hope you enjoyed today's episode of How the Body Works. Feel free to click, follow, and subscribe. Check us out on Facebook or visit our website.
All right, thank you very much. So, the one who requested for a replay, I'm sure now you're welcome. All right, so let's quickly move on to what I call the organic biomolecules. Are you okay? All right. So, a few things to note. Number one, the organic biomolecules. They are very stable. And well tolerated in our cells. And body fluids. They are very stable and they are well tolerated in our cells and also in body fluids.
Sir,
Number
please,
two, body fluids. White fluid. It's dirty. Number 2 is that they are made up of carbon, hydrogen, and what? Oxygen. They may contain other new elements like nitrogen, like sulfur, like phosphorus, or even metals. But these are the basic ones. All of them, they contain carbon, hydrogen, oxygen as their structural backbone. Structural backbone. Number 3 is that they react slowly among themselves. Therefore, they may require what? Enzymes. If you leave them to react to themselves, they react very, very slowly. So to make it very fast, they require enzymes. Example, if you have glucose combining with galactose to form lactose, this reaction occurs in the mammary gland. But if you leave it to occur by itself, it will take like forever to happen. But if you have enzymes, then it can occur quickly. So you can get lactose as breast milk for the baby. Number 4.
Please, you said glucose combining with
Galactose.
Please, take the number 3 again, please, sir.
I said they react slowly among themselves. Therefore, to make their reaction more fast, they will require what? Enzymes. They will require what? Enzymes. And I give the example. Reacting with galactose in the mammary gland to form lactose. If you leave glucose and galactose alone to react, the reaction will never. It will be very, very slow. Very, very slow. So to make it very fast, enzymes are thrown in to facilitate the process. Number 4, they exhibit stereoisomerism. Shall we all say it together?
They exhibit
stereoisomerism. So we hear the last part.
They exhibit stereoisomerism.
Stereoisomer. Wait. Okay.
Again.
Stereoisomerism.
Stereoisomerism.
So let me just show you what I mean by that. All right, please, can you all see my screen here?
Yes, sir.
Yes, sir.
Yes, please.
Okay, now you can see that. Look at the ones on the top left corner. You see some 3 carbohydrate isomers. Can you see them?
Yes, sir.
Yes, sir.
Yes.
Can you see my cursor?
Yes.
Sir. Yes, sir.
So what do we have here?
Glucose.
What is it?
Glucose.
Glucose.
Galactose.
This is what?
Fructose.
I want you to look at glucose and galactose. Look at them carefully. Are they the same?
Yes.
No,
No.
sir.
No,
They're not the
please,
same.
they are not the
Not the
same.
same.
They are not the same.
Okay,
No, they are not the same.
what's the difference? Can you tell me?
On the third straight line.
The fourth.
The fourth line.
No, the third
Okay.
straight line. The first one branches. For
Yes.
the glucose, it is HCOH, but for the galactose, it's HOCH.
Oh, that's beautiful. You have a very, very good eye. So that is it. Are you okay? And that is the only difference between glucose and galactose. It's just the rearrangement. They all have the same number of carbon atoms. Let's check the number of carbon atoms. Can we count? There's what?
1,
2,
2,
3,
3,
4,
4,
5.
5, 6.
Okay. Galactose.
1,
1,
2,
2,
3, 4,
4,
5,
5,
6.
6.
Okay. So we have 6 carbons, isn't it?
Yes, sir.
Okay. I want you to count the hydrogens. How many hydrogens do they have?
1, 2,
2, 3,
4,
4, 5, 6, 7, 8.
11.
11.
It's 11.
9.
11 for glucose.
Say 18.
Glucose is 11.
Oh, God.
1, 2,
Say 18.
3, 7, 8, 9,
So let's count again. 1,
Glucose is
2,
12.
4, 5,
6,
6, 7,
7, 8,
8, 9.
10. 11. It's 12. 11 is 12. It's 12.
12. And
It's
this is how
12.
many? 12. Now, oxygen. 1,
It's
2,
11.
4, 5, 6. So you can see that the two of them, glucose and galactose, they have how many carbons? 6. How
Yes,
many
sir.
oxygens? 6. How many hydrogens? 12. So between them, they have the same number of atoms of carbon, hydrogen, and what? Oxygen. The only difference is in what? The arrangement on carbon 4. Are you okay? So we say that they are anomers. Are you okay? Hello, please, are you with me?
No, please.
Yes,
Yes,
sir.
sir.
Sir, please come again.
The only
So
difference,
please come again.
the only difference is on the arrangement of groups around carbon 4. In the case of glucose, you have H to the left and then OH to the right. In the case of galactose, it's rather OH to the left and H to the right. Are you okay? And just this arrangement is what results in the drastic changes in the physicochemical properties of these substances. Glucose is very sweet, but galactose is very, very, it's not that sweet. Are you okay? And etc.
Is it
So
better?
this is what I was referring to as what? Stereoisomerism. In other words, they can change their arrangement and appear in different forms. Are you okay? If you consider glucose and fructose, you can see that they also have the same number of carbons, same number of hydrogen, same number of oxygen. The only difference is that fructose is what? Is a ketone sugar. Glucose is rather an aldehyde, is an aldose sugar. Are you okay? Now I want you to look at these two. What are they? This is what? L-glucose. And this is what?
D-glucose.
D glucose.
Okay. Now, what's the difference between L and D? The difference is on carbon 5.
One is
Can you see that?
yes, sir.
Yes, sir.
One is 1C.
The difference on carbon 5. In the case of yes, of L-glucose, the OH is to the left. But in the case of D-glucose, it is to the right. Now note this. Physiologically, our body is able to metabolize the D-glucose, but cannot metabolize L-glucose. I say it again. Physiologically, our body is able to metabolize D-glucose, but not L-glucose.
So why?
I don't know. So this is just to let you know that this concept of stereoisomerism is very, very important. Are you okay? So the body's enzymes are able to identify the D-glucose to the neglect of L-glucose. So many times in weight management, we tend to use L-glucose for people who are obese and diabetic because they can't metabolize the L-glucose. So you use it for diet for them, and then it just come out. Are you okay? Now let's look at these two. This is what? D-alanine, and this is what? L-alanine. I'm sure you can see the two.
Yes, sir.
Now, physiologically, the body is able to use, this is an amino acid, but these are two forms. Are you okay? The D-alanine is a mirror image of the L-alanine. Functionally, the body is able to use this L-alanine and will reject the D-alanine. So you will see that even in drug which contain a lot of amino acids, you see that the amino acids are in L forms. So you have L-lysine. Are you okay? Because the body will prefer the L-isomer. Are you okay? Than the D-isomer for amino acid. But for carbohydrates, they prefer the D-isomer to the L-isomer. Are you okay?
Yes, sir.
Okay. Okay. All right. So that's about it. Okay. So if I said they exhibit stereoisomerism, these are common examples of ways by which organic biomolecules are able to exhibit this. In fact, by virtue of this, a lot of the time when new drugs are being formulated, they have to test in which form it is very active in the human body, because the body can recognize different isomeric forms, and different isomeric forms tend to exert different effects. So some drugs are more effective in the L form, whereas others are more effective in the D form. Please, are you okay?
Sir,
Yes,
please.
sir.
Yes, sir.
Hello, sir.
Yes.
Please, what's the meaning of the L and the D?
The L stands for what we call level rotatory. It's an organic terminology. I don't want to take you in there to confuse you. For now, in all simplicity, just accept the L and the D.
Okay.
The L for what we call level rotatory. The D stands for dextrorotatory. Are you okay? And there are meanings to that. But I'm not teaching you organic chemistry. This is just biochem. And this is just an aspect of it. So let's not spend the whole day in going down that lane.
Okay, sir.
Please, are you okay?
Yes, sir.
All right. So now let's look at some common examples of biomolecules and what they do. So let's go through the list quickly. This is just a teaser. The first one is what? Shall we all say it together?
Folic acid.
Folic acid.
Okay. Folic acid.
Hyaluronic acid.
Okay, thank you.
Glutathione.
Glutathione. Acetylcholine.
Acetylcholine.
Choline.
Prostaglandin.
Prostaglandin. Glucose. Ribonucleic acid.
Ribonucleic acid.
Thank you very much. So, let me say this. Cholic acid is oftentimes a derivative of cholesterol. In fact, it is the way by which cholesterol in our body is removed. The principal means by which cholesterol within our body is removed is often in the form of what? Cholic acid. Are you okay? And
Yes.
it doesn't come out as an acid. Acids react with metals and form salts. So usually they are made to react with either taurine or glycine and form what you call taurocholate or glycocolate, which is what we call bile salt. And this bile salt is what is stored in the gallbladder. It's stored in what? In the gallbladder and released to help in the digestion of what? And released to help in the digestion of lipids in our body. So without the cholic acid, lipid digestion will be very, very difficult, if not impossible. Are you okay?
Yes, sir.
And usually they are produced by the liver. So the liver takes the cholesterol within our body, transform them into cholic acid, use them to produce what we call bile salt, and then they are stored in gallbladder. During digestion, they are released from within the body into the alimentary canal. So after they have helped with the digestion of lipids, they are not absorbed back, but they form part of stool. In fact, they help give the color to stool. That brownish, kind of yellowish color to stool is conferred by, to some extent, the bile. Are you okay?
Sir, please come again.
I'm saying that the cholic acid is often released into the alimentary canal where they help in digestion of lipids. Now, following digestion, they are not reabsorbed back. So they continue with the rest, the undigested and unabsorbed nutrients, to form the stool, or what we call feces. And they help confer the color to feces, that brownish, dark yellow kind of color, conferred on stool or conferred on feces, is because of the bile. Are you okay?
Yes, sir.
Yeah, okay.
All right. Then the next one is what? Hyaluronic acid. This is a very, very important substance. Currently, it has gained a lot of application in cosmetic surgery and skin rejuvenation. Are you okay? It's used in cosmetology for the treatment of various diseases. It is a very, very important component of almost all body fluids. So from the plasma or serum to interstitial fluid to blood and etc. In the skin, it's used to enhance, like I said, or treat what we call inflammatory growth on the skin and improve the skin coloration and its suppleness. It could also be used to treat arthritis. In fact, it's very, very important. And there are several uses of it now as per research findings. Are you okay? So that's hyaluronic acid. But we are considering this as a biomolecule because they are produced within our bodies. Are you okay? They are naturally produced. But the artificial ones, or what you call recombinant hyaluronic acid, that are being sold in the market for treatment of various cosmetic purposes. Are you okay?
Yes, sir. Yes, sir.
So let me just show you this short thing and then we'll continue with the rest. So this is, can you see my screen here? This is how hyaluronic acid is exploited cosmetologically. They can be applied topically or injected. Are you okay? Not so deep, but into the skin for treatment of various skin diseases and to meet various skin expectations. Please, are you okay?
Yes, sir.
Yes,
All right. So let's move on. The next one is what? It's glutathione.
Glutathione.
It's not glutathione, it's glutathione. Okay. Now, glutathione is also very, very important.
Oh, who is that?
Sir, please, I have a question.
Yes, please, you can ask.
Please, the hyaluronic acid,
Hyaluronic acid, it's okay.
can it be used for bleaching?
For what?
For skin bleaching.
Skin bleaching.
Yes, please.
Well, I'm not a specialized cosmetologist or dermatologist, but so I wouldn't be able to speak authoritatively about that. But I know from research findings and readings, extensive readings, that is being used for what we call skin toning, not for bleaching. I don't know if toning means bleaching, then fine. But if there's
No,
a
it's
difference,
mostly used for skin hydration.
okay. Please, are you okay?
Yes, please.
Uh-huh. So I haven't read that it's being used for bleaching, but I know it's being used to improve the complexion and the what we call rejuvenation of the skin. So normally people will go for what we call dermal injection with hyaluronic acid. Are you okay? But it's quite expensive, just for you to know. So that's that. Are you okay?
Yes, please. Thank you.
So here, what they are trying to do, if you can remember what I said earlier, they know that this is a natural compound that the body produces. Are you okay? And so they are using this, and because the body produces it, it's well tolerated. The body doesn't react to it. Are you okay? It doesn't react to it. That's why I said that they are well tolerated within body fluids and in tissue. So if you are administering it, apart from the fact that it's recombinant, if it's well formulated, the chance of reaction could be very minimal because it's a natural compound. Are you okay? Glutathione is a tripeptide. It consists of only 3 amino acids. It's a very, very short. In fact, it's considered as the shortest peptide within the body. Only 3 amino acids. So it's relatively small compared to other proteins. But it's very important. Are you okay? What does it do? Within our body, especially in our blood, sometimes through metabolic processes, we produce a lot of so during metabolic processes, a lot of toxic substances, usually named as free radicals, are produced within our cells, and they all diffuse into the bloodstream. So inside our blood, there is a collection of a lot of toxic waste materials. Some of them can be excreted through urine, but some may accumulate. Are you okay? And these attack immune cells and the red blood cells. That's the cells in the blood. They attack them very often. So one way by which the cells can be safeguarded, remember, if the white blood cells are being attacked and they are dying, the implication is that the immune system also becomes very weak. If the red blood cells are attacked and they are being destroyed, the implication is that you are becoming more anemic and you have less and less hemoglobin. And so there will be short of oxygen saturation and the individual can die of shock. So it's very important that we protect the red blood cell and the white blood cells from being destroyed by these free radicals. The principal agents that are able to fight these free radicals are often called antioxidants. They are called what? Antioxidants. Inside our blood, there are several antioxidants. Inside our blood, There are about 3 enzymes that act as antioxidants to fight against these toxins. Number 1 is what? Catalase. Number 2 is what you call superoxide dismutase. Number 3 is what we call peroxidase. Now, these are all enzymes and they are natural, they are within our blood. What they do is that when these free radicals or toxic substances are released and they flood the blood, then these ones will go in there and quench them and destroy them so that they don't harm our red blood cells and the white blood cells. But sometimes these free radicals can be so much that they overwhelm these enzymes. And that is where glutathione comes in handy. So the glutathione will react with these free radicals and destroy them so that it can protect the red blood cell. In fact, it has been found from research that the toxic effect of some of these substances is what causes cancer, aging, coronary artery diseases, and etc. So a lot of these free radicals have been implicated in what we call inflammatory diseases. Are you okay? Such as cancers, aging. Aging is not a disease per se, but it's been known to be promoted or accelerated by these free radical reactions. And then coronary artery diseases, usually by stimulating arteriosclerosis. So taking glutathione or having a lot of glutathione can help destroy these ones and reduce the risk of coronary artery disease, cancer, and the slowing of aging. So again, exogenous glutathione is also being used currently for the management of aging. So it's also being exploited cosmetologically. Are you okay? To slow down aging and maintain youthfulness of the skin. What they call the youthful glow of the skin. So that's about it. In fact, a lot of these biomolecules are being exploited through what we call cosmetic technology. So many of them are now being synthesized in the lab and used for treating various cosmetic or for improving general skin health and cosmetic outlook. Yeah, Victoria, your hand is up.
Please, I want to find out, can someone with leukemia use the glutathione?
Someone with what?
Because Max leukemia. Max International developed this kind of glutathione. It's in the form of a capsules.
Yes.
So can it still help fight the disease? Okay, thank you.
Okay, this is what I would say from my experience in biomedicine. You can use it as a treatment, but to some extent, you can use it to manage. Are you okay? Because the individual, the idea is that a lot of the red blood cells are dying because of the cancer. But the glutathione doesn't really attack the cancer. It doesn't attack the gene or the proteins in the cells that causing it to die. But it's rather attacking the free radical. And much as you don't know if there's cancer because of oxidative stress, taking the glutathione wouldn't really mean much. At best, it can only help eliminate possible complications or acceleration of the cancer. But you can use it to treat.
Okay.
So that's what I can say. Please, are you okay? Victoria, are you okay now?
Yes, sir.
All right, thank you. So that's basically about glutathione. The next one was what? Acetylcholine. Acetylcholine is a very, very vital substance or biomolecule. It's important in what? Nerve impulse transmission. Without acetylcholine, nerve impulse transmission would be a bit difficult. Of course, there are several nerves. We have what we call adrenergic nerves and cholinergic nerves. Are you okay? But there are more cholinergic nerves. The cholinergic nerves require acetylcholine for impulse transmission.
Sir, please, can you repeat what you said again?
I said that in terms of nerve impulse transmission, there are two types of nerves. We have what we call cholinergic nerves and adrenergic nerves. Are you okay? The cholinergic nerves require acetylcholine for impulse transmission. So without them, impulse transmission becomes very All right, so let's kindly watch this short video. Let's see.
This storage, release, receptor binding, and degradation. And what are some clinical correlations associated with acetylcholine? Hello, everyone. My name is Dr. Morton, and I'm the noted anatomist. First question, what is acetylcholine? Well, it's a neurotransmitter derived from acetyl-CoA and choline. So there's the chemical structure, there's the acetyl group, there's the choline group, and shing, they make a wonder twin power of acetylcholine. And there's the ACH abbreviation. Now, many neurons release acetylcholine. For example, all parasympathetic neurons, both preganglionic and postganglionic parasympathetic neurons, release acetylcholine. Sympathetic neurons do, but only the preganglionic, not the postganglionic, with the exception of sweat glands. And then all somatic motor neurons.
One thing I keep telling everyone is you don't need to japa to live a certain life. Man, it's freaking cold today. I'm just heading out to the office now. And one of the
release acetylcholine into the neuromuscular junction. Anywhere there's a skeletal muscle, there's a neuron that releases acetylcholine to cause it to contract. And then a variety of central nervous system neurons found in the basal forebrain, the diencephalon, the hippocampus, and so forth. Basically, all peripheral nervous system motor neurons release acetylcholine except postganglionic sympathetic neurons, but even there is one exception. The central nervous system neurons in these video tutorials are not ones that I'm going to focus on so much. The following steps comprise the process of acetylcholine transmission, and I'm going to talk about steps A through E. We're going to talk about a clinical correlate with individual vesicles, like that. So basically, VACHT loads the gumball machine. If the gumballs are acetylcholine, it puts them all together into a closed space. There's about 1,000 to 50,000 acetylcholine molecules per vesicle. So it doesn't look so much like this picture as it does that picture. But then there's also 300,000 vesicles per cholinergic neuron, so it doesn't look so much like that as it looks like that. And so in this EM, the letter T represents an axon terminal, the letter M a skeletal muscle, and those circles in the axon terminal are individual vesicles containing thousands of acetylcholine molecules. Now, acetylcholine-bound vesicles are protected by acetylcholine esterase, which is an enzyme that cleaves acetylcholine into its acetate and choline counterparts. But if you have acetylcholine inside the vesicles, acetylcholine esterase cannot degrade them. So there is the storage of acetylcholine. Now, a clinical correlate, there's a drug called vesamicol that blocks the vesicular acetylcholine transporter. When that occurs, it inhibits acetylcholine from being stored in a vesicle. And these empty vesicles that have no ACH, then when the neuron goes to secrete them, that vesicle fuses with the membrane and there's little to no release of acetylcholine. Let's now talk about the release of acetylcholine. An action potential causes an influx of calcium and promotes vesicle fusion with the membrane, and then ACH is released into the synaptic cleft. So here's a terminal axon, and an action potential spreads over the terminal axon, and then it causes this voltage-gated calcium channels to open, and calcium influxes from a high to a low concentration into the cytoplasm, and then the calcium interacts with the vesicles, causing them to fuse with the presynaptic membrane via these snare proteins. And snare proteins mediate the fusion of the vesicles with the presynaptic membrane for release of acetylcholine into the synapse. In other words, you put nickels into the gumball machine to get gumballs out. And so similarly, you put calcium into this terminal axon to get acetylcholine out. Unless you have a condition called Lambert-Eaton myasthenic syndrome, which is a rare autoimmune disorder where antibodies block and destroy voltage-gated calcium channels. When this occurs, calcium cannot enter into the cell, and therefore calcium-dependent triggering synaptic vesicle does not occur. And then as a result, acetylcholine vesicles do not fuse with the membrane, and the neuron does not release acetylcholine. Another clinical correlate is this botulinum toxin, which cleaves the snare protein complex. Without the snare proteins, the vesicles cannot fuse with the membrane, and as a result, neurons cannot release these vesicle molecules. The botulinum toxin results in a disease called botulism, the most poisonous biological substance known. And it's Latin for botulus, which means sausage, because it was called the sausage disease because pork sausage particularly is what people realize. If you had bad pork sausage, you get really, really sick, and a good chunk of people die. This physician named Justinus Kerner was ahead of his time. He was the first physician in Germany and really recorded to describe what we now know as botulism, and he predicted that someday the substance causing the disease would have medical applications. So the botulinum toxin in the early 70s was used. They took a dilated version of the botulinum toxin and they treated strabismus. So they would inject it into the lateral rectus. In this case, this patient has the lateral rectus is too tight and it paralyzes some of the strands of the lateral rectus to relax the eye to its natural position. Hyperhidrosis, where people are sweating in the palms of their hands like crazy, you inject the botulinum toxin, it stops those neurons from secreting acetylcholine, they stop sweating. Overactive bladder, when there's always this urgency to urinate, you put 3 or 4 of these injections of the diluted botulinum toxin into the bladder, and it relaxes causes the bladder so it's not contracting. And then cosmetically, for wrinkles and also for migraine headaches, is what the botulinum toxin is used. It's a beautiful example of human ingenuity, why they call it the miracle toxin.
Guess their age.
I'm 38.
I'm 50.
I'm 68.
We're doing this face yoga plan to look.
So there's the synthesis, storage, and release of acetylcholine. Let's now talk about cholinergic receptors. So acetylcholine interacts with 2 main classifications of cholinergic receptors, nicotinic and muscarinic. Squirrel! I'm going to take a bit of a tangent here for a second. So there's a tobacco plant that makes a molecule called nicotine, and scientists realized that nicotine binds to receptors inside the body, and they said, hey guys, what do we call this receptor that binds nicotine? And they said, I know, let's call it nicotinic receptors. Then another group said, hey, there's this mushroom that creates a molecule called muscarine, and muscarine binds to receptors inside the body. And they said, gentle people, what do we call these receptors that bind muscarine? They said, I know, we'll call them muscarinic receptors, hence how we have nicotinic and muscarinic receptors. Something I want you to realize is that both nicotine and muscarine are not made in the body. They're exogenous molecules. But nicotinic and muscarinic receptors are made and found in the body. So the pockets of the nicotinic and muscarinic receptors are specific to nicotine and muscarine respectively. So nicotine cannot bind to muscarinic receptors and muscarine cannot bind into the pocket of a nicotinic receptor. Now enter acetylcholine. Acetylcholine is made and found inside the body, and the structure of acetylcholine enables it to bind within the pockets of both the nicotinic receptors and muscarinic receptors. So cholinergic receptors of nicotinic and muscarinic. So the take-home message is this. Acetylcholine binds to 2 types of cholinergic receptors, nicotinic and muscarinic. Okay? Now the structure of acetylcholine is not really the shape of a circle and a square. I wanted to, I drew it this way to show that it can bind to both of the pockets of the nicotinic and muscarinic receptors, and that's the physiological and pharmacological thing I wanted you to recognize with acetylcholine. Now, there are a couple of different types of nicotinic receptors. There's nicotinic receptors found only on skeletal muscle, hence the N, little m for muscle, and also found on postganglionic neuronal cell bodies, hence the little n for neuron. You can also find nicotinic receptors on the adrenal medulla chromaffin cells. These are ligand-gated ion channels. Now, muscarinic receptor types, there's 3, there's actually 5, but we'll focus on M1 in the CNS, M2, these receptors are primarily found in the heart, and M3, which is found everywhere else in the body where parasympathetic innervation occurs, plus sweat glands. And muscarinic receptors function through a G protein coupled receptor where they use second messengers. There.
All right. All right. All right. So this is just an illustration of the physiological and the clinical utility of what we call acetylcholine. Are you okay? It's a very, very important neurotransmitter produced by our various nerve cells, are you okay, at the presynaptic and postganglionic spaces to enhance neurotransmission. So this is very, very important. All right, the next one was glucose. And glucose coming as a ready fuel to generate energy for the body. Then the last one was what? Ribose sugar, which is used to produce what we call ribonucleic acid, are you okay, or RNA, which help in gene expression. Please, are you okay?
Yes, sir.
Yes,
All
sir.
right. All right. So at this moment, let me pause and ask if anybody has a question.
Sir, the last one.
Oh, ribose sugar. Please, any question?
Hello, sir.
Yes.
Sir, please, can you please repeat with the colic acid? I got confused at a point, and my network too was misbehaving.
No, I said that the colic acid is a derivative, the compound generated from what? Cholesterol. It's a compound generated from cholesterol. In fact, that's where it gets its name. Are you okay? And usually it's not released as an acid, but rather it's made to combine with another compound to form what we call a salt. That's why we often call it bile salt. And they are released into the intestinal lumen to help in the digestion of lipids. And I said that following the digestion, they are not reabsorbed. So they flow down with, number one, undigested food and unabsorbed nutrients, which collectively make up what we call the stool or the feces. So the bile salt, apart from helping in the digestion of lipids, in fact, the truth is that in all simplicity, it causes the lipids to foam up. Are you okay? We call it micellization. It causes the lipids to break up into small droplets. So, I mean, grossly, we will say that it causes the lipids to foam up so that they can have large surface area. And that allows the enzymes to be able to break them down quickly. But apart from that, in the stool, they add color. Are you okay? They add color to the stool. That brown, dark yellow kind of coloration is because of the bile salt. Lady, are you okay?
Yes, please. Thank you.
You're welcome. All right, so I don't think I have ample time to start with the carbohydrates. So I just want to end it here today. Thank you all for coming. May the good Lord bless and keep you. May He cause His face to shine upon you.
Amen.
Amen.
Amen. God bless
May
you.
Thank you. Direct your path.
Amen. Amen.
Jesus will pray.
Amen.
God bless you.
All right.
Amen. Yes. God
God
bless you.
that will all pass. Mm-hmm.
Amen. Amen. God bless you, Reverend.
Pastor Jenna say I can. You be my network na enyo.
Na me pacho slice na ya be dan na saka.
Says e wo ho dada.
Hey. E wo group no mu.
E wo group no.
Yeah, ya
Ah,
join
yeah.
nche ya.
Financial.
Pastor last
Please,
part
Pastor,
no last part no then na say I can. Na your network
no
new.
network new. No bomb pie one or bomb pie one.
Oh, okay, thank you.
Why should I have left?
Please call first. I joined the group later. How will I get the slide?
The slides are already in the group, so